a privilege to hear from Dr. Capecci on his history. And I'll focus on the work that we do with stem cells. So I have like a dual hat here because I'm a researcher, but I'm also a parent of someone with autism. This beautiful boy here is Ivan. He's a 17-year-old, and he has profound autism. And that's also a reminder for me to say that when we talk about autism, I always like to classify what kind of autism we are talking about. Since I'll be talking about treatment, therapies, gene therapy, I'm not dealing with autism level 1 or 2. I'm dealing with autism level 3, which are the most dependent, the most vulnerable population, the ones that needs treatment. I think, we heard a lot from the geneticists about how genes are important for autism, but the critical thing here is how to understand the role of those genes. And we hear about animal models as well. But early in my career, I realized that mutations in some of these autism genes don't always translate in phenotypes in mice, and I was looking for a human model for autism, and especially because understanding how autism grows up in uterus to develop like a functional brain was also part of my curiosity. When I started my lab, I was highly impressed by this publication from Yoshiki Sasai from Japan, showing that you can neuralize human pluripotent stem cells in 3 dimensions to form structures that are quite similar to the human brain. And for those who attended the early version today on the round table, both Georgia and Alex, they did a great job explaining about this technology. I'll just remind you of the limitations because you're going to hear a lot in the media about mini brains in a dish, and it might give you, especially for the parents, the false impression that you have a fully formed human brain inside the lab, and that's not the case. I'll show you in a minute how they look like. But some of the limitations, including the size of the tissue, the lack of vascularization, the immaturity of these neurons, and down the road we might have bumps into the ethical and moral issues. There's lots of protocols out there. I'll be focused on the protocol that we develop in the lab. We like it to call it a semi-guided protocol. This is because we give like an initial push towards the cortex, but we also allowed for other brain regions to be involved. And we start with single cells, and after induction of proliferation and maturation, we end up with spheres that contains about 5 million cells in there, 2.5 million neurons, and the other cell types are mostly glia cells. So this is a postdoc in the lab handling a plate of a single batch of organoid that comes from a 6-centimeter iPSCs. And as you can see, they grow and they become visible at naked eye, and you can make thousands of those at the same time. Actually, at any time in my lab, if you come and visit, you're all welcome to do it. There's probably 100,000 of these organoids from different genetic alterations in our incubators. So they self-assemble, which means that the tissue will be formed by the genetic instructions that they contain. So you can capture all the genome of the person in these pluripotent stem cells, and they form like ventricles like this one, and you can see the projections of the radioglia cells that actually helps on the migration of these neuroprogenitor cells to form the cortical plate. It's not perfect, and we don't always have the representation of all the cell types, and this is a question that I get a lot, what kind of cells you have in there? Well, it really depends on what stage we are talking about. This is a snapshot at 4 months, where we have the pool of progenitor cells in there producing mostly glutamatergic neurons. At that stage, we also see that the pool start to produce astrocytes, here in red, and we start to see a discrete population of neurons presenting GABAergic markers. They are not yet inhibitory markers. They will become inhibitory neurons down the road. So what's relevant is how to use this model to really push towards therapies for autism. And what my lab has been doing is collecting a series of subtypes of autism, mostly classified based on the genetics, to really find phenotypes in addition, then test therapeutics, and perhaps move into clinical trials. I'll give you like some examples. This is for a subtype of autism that has mutation in TCF4, also known as Pitt-Hopkins syndrome. Very severe type of autism. Most of them are non-verbal, have seizures, severe constipation, lack of motor activity, they will eventually end up in a wheelchair. Not much we can do, but this is caused by haploinsufficiency, meaning that 50% of the protein is reduced on the brain cells of those kids. What if we use like a gene therapy approach to actually bump it up, the level of protein? There is only one caveat. Most of these neurodevelopmental genes, if they have too much expression, is as bad as if they don't have enough expression. So we have to be creative on the genetic tools to kind of modulate the level of expression. And this is the work from Fabio Papis in the lab. And his idea was to clone these E boxes, which is the sequence of the transcription factor, the TCF4 that binds upstream to a minimal promoter to have a feedback loop that self-regulates the amount of protein produced in each cell type. And you can see in the Western blot over there, depending on the number of repeats that we clone upstream to the gene, we can modulate the level of expression. As soon as we find the correct level of expression, we put that in an AAV vector, and we use to transfect the brain organoids. And not only we could correct the expression of TCF4 to the same level as controls, but we also correct all the downstream genes that are regulated by TCF4. Most importantly, what we observe is that in the mutant organoids in the middle, we have morphological alterations, for example, lack of these rosettes, that reduces some of the progenitor cells, that will actually reduces also some population of cortical neurons such as CTP2. Upon correction of the viruses, we can not only restore the gene expression, but we can start to see some of the population of neurons that were missing, and also the structure of the organoid becomes close to the neurotypicals, which is quite amazing to see. Happy to say that this technology was licensed by a company called Mazi, who is taking this to a clinical trial. Another one that is moving to clinical trial is with CDKL5. This is in collaboration with a company called Ultragenics. CDKL5, if you have a mutation in CDKL5, you are born seizing. Most of these are intractable seizures. And what we observe in the gray bar compared to the black bar is that the mutant cells have, these neuronal cells have hyperexcitability, most likely making the brain susceptible to seizures. With the gene therapy, we could push down the hyperexcitability of these cells. So we are accumulating more and more evidence of what we call proof of principle, and we are getting like a pipeline to test some of these reversibility phenotypes. So we recently received a large donation to create a gene therapy initiative at UC San Diego together with my colleague Stephanie Shirky. We are moving in that direction. So you might say that's great if you know the gene, but most of the autistic individuals are polygenic. We don't always know the gene. So that's why we decided to turn into studying the network inside of these organoids. And while it's clear that if you patch a neuron, you're going to see like some activity, what was unclear to us if they were forming the right connections inside of these organoids. And these are examples of things that you might be missing if you're just paying attention to one person. There's a whole thing going on inside the stadium here. So the way to do that without disentangle or destroying the organoid is to plate them on top of multi-electrode arrays. These are electrodes that are plated in the bottom of the dish. You just keep the organoids in there and you record the activity over time. And to our surprise, what we found is that not only the electrical activity starts to increase, but when they reach about 4 months of age, they start generating what we call neuro-oscillation, suggesting that different parts of the organoids were starting to communicate with each other. And these oscillations becomes more and more complex as we age the organoids. So a question came, which is how similar are those neuro-oscillations to the human brain? So why this is an important question? Because if you can compare the neuro-oscillations in the organoid with an EEG, we have a bridge, or the first step towards a bridge between the basic sciences and more cognitive function. So we did that with machine learning by training a machine to recognize the age of the subject based on the EEG, and then we finally provide the machine with the data from the organoid, and we ask in an unbiased fashion, can the machine predict correctly the age of the organoid? And this is the data. The predicted age is one axis, the actual age is in the other one, the perfect prediction is in the dotted line. The human brain data is in black, and the organoid data is in blue. So each one of these dots is one organoid. And I hope you can appreciate that while they are quite variable, they follow the same trajectory. This is highly significative, which means that the machine gets confused. It cannot distinguish between the organoid or the human brain based on their electrical activity up to 40 weeks or 9 months of age. So then what we see, which was interesting, is that even though we can keep these organoids for several years in the lab, the electrical activity plateaus to a point. And we feel that what was missing is some kind of an input and output. And we were trying to do that by looking at the responses of the organoid upon stimulation. The setting is something similar to what is on the top here. You have an organoid that is being stimulated by optogenetic or electrical stimulated while we record from the other side. So we can actually stimulate and record from the same side. And we play around with different stimulus to see what kind of response we get from the organoids, hoping to have this sigmoid curve here, and that's exactly what happens. So there is a level of stimulation that they respond really well, but more or less they might not. So we decided to look for evidence of plasticity inside of these organoids. And in neurosciences, synaptic plasticity is basically demonstrated by LTP or LTD, which is just like terms to suggest that the organoids are either eliminating unnecessary plasticity or making eliminating the unnecessary synapses or making the relevant synapses strong. So the idea was to give like a stimulus to the organoid, record a little bit, and then to see if after an hour or so, if they continue to remember that stimulus upon like a short stimulus, just to see if there is any memory being recorded inside of these organoids. And what we have is something quite interesting. So what you have here is the response from the different electrodes in the different wells inside of that carry these organoids. If it's white, it's because the organoid is not responding. If it's blue, it's because it's responding less. If it's red, it's because it's responding more than what it's supposed to do. So what does it mean? So this means that they have both LTP and LTD in the same organoids. They are either making synapses stronger, but also eliminating unnecessary synapses as they mature. And this is quite important to know, because if they do have this mechanism, they are able to store, for example, memories or even to learn something. And we are trying to harvest the biology to see if we can teach these organoids to do something. And we are starting with the visual cortex, since this is one of the sensory informations that is implicated in autism as well. And the way we do that is by creating, using the same iPSCs, both retina, thalamus, and the cortex. We put them together, and we can visualize the photoreceptors in green, the trajectory of the axons passing through the thalamus and going to the cortex here in red. So this is a picture of how they look like after a little bit of maturation. We start to see the formation of the photoreceptors, and what we're trying to do now is to visually exposing these organoids to different stimulus and see if we can remember that after an hour or so. So basically creating visual memory in these organoids. Once we have that, we can try with different subtypes of autism. So another way to do the same thing is to have like a stimulation that takes the entire body. So what you have here is a robotic platform that is interfacing with an organoid that is sitting inside of the incubator. The electrodes capture the electrical activity and send to a computer that decodes the connections between these synapses and send a message to the robot and learn how to walk, how to move the forelegs. We are just adding now the sensory information into the robot. These eyes are infrared that will tell or will stimulate the organoids as it gets close to an obstacle. As it stimulates the organoid, the network changes because it has the plasticity. A computer can sense that and send a second command to the robot, like turn left or turn right. Again, I mean, we can use this platform to teach the robot how to navigate a maze and see if they can remember all the necessary steps without the help of the computer. Once we have that, we can repeat the same experiments with autism spectrum disorders or intellectual disability conditions that we know have more difficult time on learning and memory. So another way that we start more recently to mature the neurons was to cultivate these brain organoids at the International Space Station. So you might ask, why he's doing that? So this was kind of inspired by a project from NASA called the NASA Twins experiment, where genetically identical twins were compared. One of them stay at the International Space Station for about a year. The other one stay here on Earth. After they return from orbit, they compare the physiology. And there are many things that changes. For example, the astronaut become 10 inches higher than his brother. There is a density of bones and muscular mass. This all changes while in orbit. But to me, what's important is this data from the publication showing cognitive scores on both of them, pre-flight, during flight, and post-flight. And I hope you can appreciate that post-flight, the astronaut, which is the blue line, has severe cognitive decline compared to his brother, and even to the group of controls. So this is something quite important. It suggests that the space environment is doing something to the brain that we cannot actually study because we don't have the brains from the astronauts. So one idea was to send the organoids using this autonomous platform. So we basically grow them in tubes, and there is a whole microgravity platform that is fully autonomous. And this is just to eliminate any action from the astronaut. The only thing that they need to do is to plug the machine inside the cabinet in the background, and we'll have full control of the experiments running this shoebox. So we kept the organoids there for about 30 days, and then we bring them back. And what we learned is that they have accelerated aging. So the mechanism why this happens is still unclear, but both by gene expression, protein expression, we do see that there's lots of pathway related to accelerated aging. We are testing now on a more physiological experiments if that's true at the functional level or not. We can even compare our data set with the NASA Twins experiments and find driver genes of these kind of senescence phenotypes by contrasting with our own data. So in one of those missions, we had like several so far. In one of these missions, we also sent brain organoids from Rett syndrome girls. I mean, these are one of the most severe syndromic forms of autism that has mutations in the MECP2 gene. And again, it's a dramatic condition. And what we notice is that by sending the organoids from the Rett syndrome patients there, we see release or pathways indicating a viral infection in these cells. So basically, the brain organoids were responding in this genetic background as they were infected by a virus. And that also indicates that they might have like some inflammation, and we measure levels of IL-6 and other cytokines, suggesting that they are highly inflamed. What kind of cells in the organoids might create this issue? And we thought that astrocytes are probably the culprit because Gail Mandel here has shown that if you mutate MECP2 in astrocytes, you basically recapitulate the phenotypes. So there is a non-cell autonomous component coming from the astrocyte that actually dictates how the neurons work in Rett syndrome. So we look for cytokines from Rett syndrome patients in vitro, and it turns out that, yeah, they are highly inflamed. They have high levels of IL-6. If you CRISPR-corrected the mutation, the IL-6 level goes down. We also validated in postmortem tissue. We can see evidence of overexpression of IL-6 in postmortem brain tissues compared to controls. So this is all very interesting, but we never infected these astrocytes with any virus. So how come they are releasing this inflammatory pathway against the viruses? And that brings me back to one of the studies that I participated when I was a postdoc, showing that MECP2, the gene that is mutated in Rett syndrome, controls the expression of endogenous retroviruses in the cells. And if you're not familiar with endogenous retrovirus, they are basically the vast majority of your genome. 20% of them are composed by line elements, or L1. These are 20% of the genome that are still able to mobilize because they are self-autonomous, and you can actually visualize that by tagging a reporter gene, the EGFP, inside one of these elements, and all the time when they are released from the genome, you can actually visualize by the fluorescent green. We also made like an entire animal with this reporter gene, and this is in the two isogenic backgrounds here, and each one of these green dots is one of the brain cells that contains at least one round of retrotransposition. And I highlighted here some of the brain regions that are more affected in Rett syndrome compared to controls. So basically what I'm saying is that MECP2, when mutated, is unleashing line expression from the genome, and while it's unleashed these endogenous retroviruses, it's accumulated in the cell, causing the false impression that the cell is being attacked by a virus. So if this is all true, we should be able to at least ameliorate the diseases by treating the cells with antiretroviral drugs, and that's exactly what we're showing here. There are several phenotypes related to inflammation, high expression of ROS or glutathione. We can reduce that by treating with HIV drugs. The same is true for the size and morphology of the neurons inside of the organoids. You can rescue that by exposing them to reverse transcriptase inhibitors or HIV drugs. We can also take advantage of the mouse model because in MECP2 they're quite dramatic. They die after 4 weeks. And by treating them with the antiretrovirus, we can not only improve their survival, but improve their quality of life. We can even perform like some cognitive tests or motor tests. And in all these tests, I mean, the ones that were treated with the antiretrovirus can kind of improve, which is amazing. So we look at the cellular level to see if we are restoring phenotypes at the cellular level. And one of the most dramatic phenotypes is the morphology of the neurons. In Rett syndrome knockout cells, the soma size is tiny, quite small compared to controls. But after the treatment, they become the same size as the Y-type counterpart. The same is true for the spine density. In MECP2, the spines are more rare or less frequent. But after the treatment, you can actually restore the neuronal morphology. So we are so excited with that that I'm taking that to a clinical trial. Just full disclosure, I'm a co-founder of Tismo. This is a parent-based company who are looking to develop treatments on non-conventional ways. So when I told them about this result, I mean, we all get excited to see if we can move that to a clinical trial. So the space project is actually revealing us some novel pathways that we couldn't see here on Earth. So that's a quite strong observation, or very appealing. And I'm happy to say that last year we founded the Astro Biotechnology Hub here at UCSD, really to attract people or partners on the space industry to see if we can create like a stem cell lab at the International Space Station and reproduce most of these data with other conditions. We are also looking to explore different types of treatments that are less conventional. And one thing that I'm recently involved it is to use the biodiversity from the Amazon. So last year we made like an agreement between UCSD and University of Amazon where we can together explore the biodiversity of the Amazon in some of these conditions. So why I'm forcing that? Because 40% of all the drugs that are out there actually come from plants. And 20 of the top 100 blockbuster drugs is actually plant-based. So there's lots in plants that we ignore. And when I was talking to some of these indigenous tribes, they tell me that there are so many of those species that have neuroactive components that we just ignore. So our goal is really to engage this community. And I'll briefly show like a video here with my colleagues from the Amazon. So engage these communities to help us to search for these neuroactive molecules. They are all spread out in the Amazon, sometimes not so easy to find. So what we do is we parachute these robots that contain sensors that we use the tribes to learn the different plants, and we can then, rescue them and rescue the plants and bring back to the lab to isolate the neuroactives. We do that directly in the Amazon. And the idea now is to use this kind of a microfluidic platform where we can test the drugs directly into the organoids that are in orbit. So this is research going on. We are using all optical electrophysiology to kind of see the neuromodulation inside of these organoids. So I will stop here. I'll thank like members of my lab. These are like quite broad. There are so many people that work on these projects and my funding agencies. And I'll be happy to take any comments and questions. Thank you.
Thank you, Dr. Moutri. Do you have any questions?
I'm very curious about the nature of the clock in the, as you age the neurospheres and culture over about 8 months. What is the clock that is driving it? And say if you made a Layman B mutation like progeria or something, do they age faster? Because individual neurons don't have that kind of an aging clock. This is something that is coming out as a function of the neurosphere.
Yeah. So definitely if you mutate genes in progeria or others, you can